Depending on the cell type, the frequency of cell division will vary. For embryonic cells, the cell cycle is less than 20 minutes. For skin cells, it's between 12 and 24 hours. For liver cells, they retain the ability to divide, but they keep it in reserve. They typically will not divide unless they are damaged, the tissue is damaged overall. And then when they are not dividing, they stay in a G0 state. The G0 state is the non-dividing working state of a cell. At any given time, most cells are in G0. And then there's some cells that are permanently in G0. These include mature nerve cells and muscle cells. These are cells that once they're mature, they'll never divide. Not all cell types have the same cell cycle. Therefore, a multicellular organism needs to coordinate cell division across different tissues and organs. This is done through cyclins. Cyclins are proteins that regulate the cell cycle. What they will do is they will bind to CDKs, or cyclin-dependent kinases, which are a type of enzyme, and this will trigger cell cycle activities. So here we have our cell cycle. Interphase includes G1, S, and G2, followed by mitosis and cytokinesis. And on this model, you could see that there are different traffic lights to denote that these are spots where the cell cycle needs to be able to go past these checkpoints if they are going to move on to the next phase. Just like you need to stop at a traffic light or a stop sign if you're going to be able to cross a road. You need to be able to stop at these checkpoints of the cell cycle if you're going to move into the next phase. This is the mechanism for how cyclin-dependent kinases work. You can see that we have the CDK, that is the enzyme, and then the cyclin, which is another protein, will bind to the CDK, and that will either activate or inactivate the complex. So you can think of it as like a triggering mechanism. There are different checkpoints along the way in the cell cycle. There's the G1 checkpoint, named the G1 checkpoint because it takes place during G1, and this initiates the DNA replication. So it asks, can the DNA synthesis begin at this point? And this is one of the most significant points along the way. Therefore, it's dubbed the restriction point. It can either restrict a cell and arrest it from going and replicating, or it can allow it for it to pass and move on to the synthesis stage, where it can start and start creating the DNA, replicating the DNA. So the S checkpoint, this is towards the end of the S phase, the synthesis phase, and it's going to initiate the protein synthesis of G2. The G2 checkpoint, this initiates the nuclear division in mitosis. So this is asking the question, has the DNA synthesis completed correctly? If it has, go ahead and separate the nucleus. And then there's the M checkpoint. This is called the M checkpoint because it takes place during mitosis starting with M, and this initiates anaphase. So you have prophase, metaphase, and the anaphase is where the chromosomes are ripped apart. So the sister chromatids are ripped apart, separated at their centromere. So this checkpoint, the M checkpoint, has to happen before anaphase can begin. So it's checking for whether the chromosomes are attached properly to the spindle apparatus, and then are the sister chromatids able to separate correctly. Different types of cyclin will show them or will express during different types or different times of the cell cycle. So this shows us the concentration versus the different phases of the cell cycle. So we can see that cyclin D is a type of protein that's going to be prominent throughout the entire cell cycle. It will peak in S and G2 phase, but it will begin to increase in concentration in G1, gap1, and it will decrease throughout mitosis. And then there are other cyclins, E, A, and B, which are specific to each part of the cell cycle. So you can see that cyclin E is going to moderate or restrict between gap1 and S phase. So it's what's going to be able to initiate the S phase, whereas cyclin A is going to be responsible for initiating mitosis out of the G2 phase, and cyclin B is going to help with initiating anaphase. So they're called cyclins for a reason, because they are cyclin. So they cycle in and out of prominence. They're going to rise in concentration and then they're going to fall. So it's a cycle. If the cell cycle is not regulated properly, then you could have an abnormal cell cycle. In other words, cancer. Cancer is an uncontrolled growth and division of cells. This is when cells spend very little time in interphase. As you know, interphase should be the longest part of the cell cycle because it includes G1, S, and G2. They're called gaps for a reason because they take a very long time. Most cells at any given time will be in interphase in one of these gaps, or perhaps in a permanent state of non-dividing, gap 0, G0. Cancer is when these gaps they're kind of being skipped. So cancer cells can kill an organism in a number of ways. They could invade surrounding cells, resulting in the loss of tissue function, or they could spread throughout the body as well. So what do we call it when a cancer spreads to other parts of the body? We call it metastasis. So causes of cancer, we have mutations to genes that regulate the cell cycle. So these cyclins are not going to come into prominence, so they're not going to rise in concentration when they're needed, and they're not going to be able to restrict the cell cycle if a problem arises. Or this cancer can be caused by genes that repair the DNA. So you have damaged DNA, and then the gene is supposed to be repaired, but it's not. So there are 4 main causes of these mutations. The first one is environmental factors. This could be UV radiation, perhaps like from the sun, UVA, UVB rays. This could be excessive heat. This could be pollution, like air pollution. Genetics can cause cancer as well. Only about 15% of cancers are caused by genetics. Another way that cancer can be caused is through carcinogens. This is when you have a substance or an agent that causes cancer, whether this is chemical exposure, radiation exposure, cigarette smoke, pollution. These are all considered carcinogens. So carcinogen means to generate cancer. And then also age can also be a big contributor to cancer as well. There is a gene called the p53 gene. The p53 gene is able to moderate the cell cycle, and what it will do is the cell cycle will be going, and if the DNA is damaged, the p53 can either tell the cell cycle to stop, in which case the DNA will be repaired, and then the cell cycle can restart. Or if it's bad enough, the p53 gene will signal the cell to undergo apoptosis, which is programmed cell death or cell suicide, where the cell will kind of just turn in on itself and the lysosomes will digest the entire cell into its components. So the p53 gene is responsible for moderating the cell cycle. So it's either going to repair the DNA or undergo apoptosis. But sometimes the p53 gene can be damaged. And if it is, you can have a problem occur in the cell cycle, and the cell cycle will just continue. And that is obviously very problematic, and it can lead to cancer. p53 can also force a cell to go into G0, resting stage, gap 0, and this will kind of just make sure that the cell is allowed to keep on persisting, but it will never replicate. So if a cancer is going to be able to become a cancer and cause problems, it has to be able to shut down that p53 gene activity. So tumors, they're masses of abnormal cells. You could have 2 different types of tumors. You could have benign tumors and you could have malignant tumors. Benign tumors, they're abnormal cells that remain at the original site as a lump. This is because p53 has halted the cell divisions. Most of these benign tumors do not cause serious problems and can be removed through surgery. The bad type of tumor, the malignant tumors, are the ones that cause problems. These are when cells can leave the original site, and they could lose attachment to nearby cells, and then the blood and the lymph can carry these cancerous cells to other tissues, and then it's going to spread. So this can then initiate more tumors in different parts of the body, and this is called metastasis. This impairs functions of organs throughout the body, and this is usually going to be deadly. There's different ways to treat cancers. You can use high energy radiation. This kills rapidly dividing cells. You could also use chemotherapy, which uses chemicals, thus the prefix chemo, to stop DNA replication, stop mitosis, stop cytokinesis, stop blood vessel growth. You could use a variety of different chemicals that are going to do different things depending on the type of cancer. A little bit on apoptosis. We looked at apoptosis on the p53 gene slide. Well, apoptosis is the programmed cell death. So what happens is the cells shrink and shrivel in a controlled process. This naturally occurs. This is a mechanism during embryonic development. For example, when the digits of your hand are forming, they're going to have a webbing between them as the the fingers form. Well, that webbing then goes away through the process of apoptosis. So apoptosis is not necessarily a bad thing, but it's a mechanism for development as well. Also, when trees drop their leaves during colder temperatures or drier conditions, they do this through a mechanism of apoptosis. So the point where the leaf is going to attach to the twig or the stem, that is going to be released through apoptosis. This also occurs when DNA is damaged beyond repair, as we previously mentioned, helping to prevent mutations and cancerous growths. There are special types of cells called stem cells. Stem cells are unspecialized cells that can develop into specialized cells under the right conditions. These were discovered in 1998 and they come in two different types. There's embryonic stem cells and then there's adult stem cells. Embryonic stem cells are pluripotent, which means they could be used in they can become different types of cells. They can be exercised into muscle cells or blood cells or nerve cells. So if you have stem cells, you can replace different types of cells that otherwise wouldn't have been able to be replaced. So we already talked about how muscle cells, they're permanently in gap 0. Nerve cells are permanently in gap 0, so they can't divide. But if we take a stem cell that is unspecialized and we specialize it to be one of those types of cells, we can then possibly replace those cells. This is a developing technology. I would encourage you to look at this animation found at this link. It's also linked on Schoology, so the embryonic stem cells animation. And then the last slide here, we have embryonic stem cells. So mitosis produces a mass of 100 to 150 cells that are unspecialized. These are the most interesting and controversial because they come from human embryos. So to take these from human embryos would mean a loss of the embryo. So studying these is obviously controversial because it results in the death of the embryo. Adult stem cells, these are found in various tissues throughout the body. They can be used to maintain and repair the same kind of tissue, but we can't specialize those cells to a different type. And that's it for cell regulation.